Welcome to part 2, lecture 2 of chapter 4, cell structure. So, chromosomes, right? Well, kind of, sort of. So here are the details or the characteristics of chromosomes for prokaryotes. Remember, prokaryotes are single-celled organisms. We call them unicellular, and they do not have a nucleus. That's the before the nucleus part, versus the eukaryote for all animals and all plants. Eu means true, karyo has to do with it, pertains to the nucleus. So these are organisms with a true nucleus. And so you can see with the prokaryote, like a bacterium, like E. coli, has a single chromosome. It's circular. Versus eukaryote, there are many chromosomes. I'll show you pictures here in a second, and they are linear, okay? Made only of DNA for prokaryotes. You have a single loop of DNA. With eukaryotes, you have DNA. They're made of chromatin. There are proteins that hold everything together. Prokaryote chromosome found in the cytoplasm, and eukaryote chromosomes found in the nucleus, specifically the nucleolus or nucleolus. And then that little last box talks about the division, cell division and subsequent division of the nuclear material. So here we see eukaryotic versus prokaryotic chromosomes. So mitosis and onion cells. So we'll talk about mitosis later, but these little dark structures here that you see, those are chromosomes. And yes, when you get to this point in cell division, the chromosomes are large enough to see with just a simple light microscope. And so you see here there are various stages of cell division going on here. Here's a nucleus, right, with a dark ring around it. There's a better one with a nucleolus or nucleolus in the middle. That's that dark spot, okay? Versus prokaryotic chromosomes, this is a single circular chromosome, okay? And this is a bacterium undergoing binary fission, which is cell division for unicellular organisms, but this is we call it the nucleoid region, but it's a single circular chromosome. All right. Okay, so with regard to prokaryotes or like bacteria, there are two domains, bacteria and archaea. Now bacteria are things like E. coli, Mycobacterium tuberculosis, okay? Archaea, archae stands for ancient. So archaea are ancient unicellular organisms, and the thinking here is that all life began as archaea. Archaea are known for, and archaea still exists, they're not extinct, but archaea are known for inhabiting things like hot springs, deep cold oceans, ice, environments that are highly saline or salinic, high salt concentrations or high sulfur concentrations. These are archaea. We call them extremophiles. They are unicellular, that means one-celled organisms. They are very simple in their organization, in their structure. They have a cytoplasm, a plasma membrane, as all cells do, and prokaryotes have a cell wall. There's one chromosome, one circular chromosome, as we have mentioned already. Some prokaryotes, some bacteria are photosynthetic, many are decomposers, and many, like Mycobacterium tuberculosis, are pathogens. Pathogens Gen comes from Genesis, which means creation. Pathos is dysfunction or disease. So anything that creates a disease or causes a disease is called a pathogen. Decomposers are obviously things that break down other organic matter. And photosynthetic, they absorb light from the sun and they make components from the sun's energy. Photosynthetic, like plants. All right, the cell wall of bacteria is made of peptidoglycan, which we'll see is different because plant cells also have a cell wall. But for prokaryotes, it's made of peptidoglycan. And penicillin interferes, antibiotic obviously, interferes with cell wall synthesis, and that's why for many bacterial infections, if you're not allergic, you will get a prescription of penicillin. Some prokaryotes have flagella, and flagella are just tail-like structures which afford the organism a means of locomotion. So here are some some micrographs of prokaryotes. Here you can see the single circular chromosome, cytoplasm on the inside. All cells have cytoplasm. Plasma membrane, all cells have a plasma membrane. You can see cell wall out here made of peptidoglycan. And the material inside, or the area inside of the chromosome, is called the nucleoid. Here we see prokaryotic organism. Now this is we call it a rod-shaped or a bacillus prokaryotic organism with a flagellum. This is a flagellum, this little tail-like structure that spins around like a propeller and allows the organism to move or propel itself through the medium. Some prokaryotic organisms may have one, some prokaryotic organisms may have more than one, and some may have flagella on this end and flagella on that end as well. So they can go back and forth, move back and forth without turning around. With regard to eukaryotes, so these are all cells in humans and large animals and fungi and plants and protists like amoeba and paramecia. These are all eukaryotic organisms. They have a true nucleus. Now, eukaryotic organisms like bacteria may be unicellular. So you're familiar probably from high school biology or 7th grade life science studying the amoeba. That's a unicellular eukaryotic organism. It's one cell, but it has a nucleus. Paramecium is that as well. Or multicellular, like all plants and animals and fungi. Complex structure, compartmentalization is what we'll see in detail as we go forth in our study. Contain the cytoplasm, plasma membrane, just like prokaryotes, but in this case we have a nucleus. True nucleus. Numerous chromosomes. Remember, prokaryotes only have one. Humans have 23 pairs, or a total of 46. Numerous organelles, which we'll look at as well, like we're talking about mitochondria and Golgi apparatus and endoplasmic reticula, and also the nucleus is an organelle. Cytoskeleton. Plants and some fungi have cell walls made of cellulose and chitin or chitin, and sperm cells are the only animal cells that have a means of locomotion by way of a flagellum. Okay. And so eukaryotes, there are 4 kingdoms. Here's your plants or plantae, your animals or animalia, fungi, and protista. We see a slipper-shaped paramecium there. So the eukaryotic nucleus is the largest organelle. It's generally centrally located, not always in all cells, but generally centrally located. It's membrane bound, and that membrane we call the nuclear envelope. And like the cell membrane, it is also a phospholipid bilayer. The nucleus contains the genetic material, which is DNA. And when the cell is not in active division, that genetic material is located inside an area that we call the nucleolus or nucleolus inside the nucleus. Okay. So human red blood cells do not have a nucleus. Many animal red blood cells do not have a nucleus. Some do have a nucleus, so you have to be careful there, but in general, mature red blood cells don't have a nucleus, and certainly mature human red blood cells don't have a nucleus. Okay, the nucleolus is where, when the cell is not actively dividing, is where you find the DNA, the genetic material. And that's also where ribosomal synthesis occurs. Ribosome is just another organelle. Nuclear pores allow passage of materials in and out of the nucleus. So we'll look at an illustration in a minute, but the nucleus is like a little ball within inside of the cell, and the nucleus, the nuclear envelope, is a phospholipid bilayer, but in that bilayer there are little holes poked in all over the place. Those little holes we call pores. So these nuclear pores allow things to flow in and out of the nucleus depending on what's needed and what can fit through there. All right, so chromatin consists of DNA and proteins. Chromatin is loose genetic material. So again, when the cell is not dividing, inside the nucleus, inside the nucleolus, is the chromatin, is just the loose strands of DNA and protein. When the cell moves into division, we call it mitosis, then the chromatin condenses and becomes chromosomes. That's when we call them chromosomes, condensed chromatin, and that's when you can see them with the light microscope. Okay, you can't really distinguish chromatin as chromatin, but when the chromatin condenses, it forms chromosomes, what you know as, or what we know as, what we'll see as chromosomes. Okay, those dark structures that I showed you earlier in the onion cells, that's condensed chromatin. We call them chromosomes. So here is the eukaryotic nucleus. Here's the nucleus proper, the nucleolus. There's the dark area between. Chromatin is all this stuff all over the place. It's just loose. It's DNA, but it's just loose strands of DNA. And you can see the nuclear, this is the nuclear envelope, phospholipid bilayer, and you can see poked into the nuclear envelope are all of these millions of tiny little pores. We call them nuclear pores. Here's a colored electron micrograph, and you can see the cytoplasm here. So this is the inside of the cell, this is the outside. There's the nuclear envelope, and you can see a little pore right there, the nuclear pore. Okay, so let's talk about some organelles. First of all, we'll start with the ribosomes. Ribosomes are sites of protein synthesis. They're found in all cells. We've referred to them as a universal organelle because you find them both in eukaryotic cells as well as in prokaryotic cells. Okay, everybody has ribosomes. Everybody has a cytoplasm, a cell membrane, and ribosomes. So for eukaryotes like you, ribosomes may be free in the cytoplasm or associated with the endoplasmic reticulum. All right, we call that the ER, endoplasmic reticulum. And so free ribosomes produce proteins for the cell. Ribosomes bound to the ER produce proteins that are transported out of the cell. So if you have ribosomes that are free in the cytoplasm, they're making proteins that the cell needs. And if these ribosomes are associated with the endoplasmic reticulum, we'll see the diagram, they're attached to the endoplasmic reticulum. We call it the rough endoplasmic reticulum, or rough ER, because those little ribosomes provide a sort of a rough texture to the otherwise smooth membrane of the ER. Okay, those ribosomes that are attached to the ER, the rough ER, produce proteins that are transported out of the cell. And we'll look at that process. You don't have to worry about the fact that the parts of the ribosome differ in Schwegberg units, but prokaryotes have 70S ribosome, eukaryotes have an 80S ribosome. It kind of makes sense because eukaryotes are much more are larger and much more complicated than prokaryotic ribosomes. All right. So here is just looking at the difference in ribosomes, prokaryotic versus eukaryotic ribosomes. So prokaryotic ribosomes ribosomes have two components, right? They have a large component and a small component, or large subunit and a small subunit. And don't worry about this. This is just a different diagram looking at the 70 versus the 80S ribosome. And I don't really care that you remember, the size of the large versus the small. I think you should remember that prokaryotic ribosomes are 70S, eukaryotic ribosomes are 80S, and they're made of two parts, a large subunit and a small subunit, because that becomes important when we start talking about translation. Okay, that's all I'll say about that. Endosymbiosis, so the symbiotic relationship in which one organism lives inside of another, where the two organisms, one inside of the other, behave as a single entity. All right, the endosymbiosis theory says that organelles like mitochondria and chloroplasts and plant cells, see, they're about the same size as a bacterium. And so the thought here is that back in ancient history, ancient geophysical history, these bacteria were engulfed by larger pseudocells or pre-cells, cells that they're not fully cells yet, but they're a structure with an enclosed membrane. And these organisms, these prokaryotic organisms, were engulfed, and the relationship is symbiotic, and the little bacterium, little prokaryotic organism inside, kind of does its thing, and whatever it does benefits the larger organism that engulfed it. And so as time went on, those little prokaryotic organisms stayed inside, and they did their thing, and the larger organism benefited. And so that's the theory. That's called the endosymbiosis, right? It's a symbiotic relationship where these little single-celled organisms were taken into these larger organisms, and given enough time, millions of years later, they're behaving as a single organism. It might make sense, chloroplasts are those structures in plant cells that carry out photosynthesis. Well, we have in modern times photosynthetic bacteria, and so the theory is that these photosynthetic bacteria were engulfed by larger cells, and they continued to do photosynthesis, making food, and the larger cell benefited from that. Okay, that's how that works. That's the endosymbiosis theory. Okay, that's a lot to say. This is a better explanation when you can see it. Here's a mitochondrion. Here's a chloroplast, mitochondrion, sites of cellular respiration, and they're not part of the endomembrane system. They have their own membrane. And chloroplasts found in plants and algae. They conduct photosynthesis, and again, not part of the endomembrane system. The endomembrane system being smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi complex. Okay, these are not part of that, and that is what leads researchers to believe that these were bacteria in the past that conducted these processes, photosynthesis and cellular respiration. They were engulfed by larger organisms and over time became symbiotic within those larger organisms. All right, another look at endosymbiosis. Mitochondria and chloroplasts have circular DNA. Did you know that? And remember that bacteria, prokaryotes, have circular DNA. Mitochondria and chloroplasts have 70S ribosomes, and remember that bacteria, prokaryotes, also have 70S ribosomes. Division for mitochondria and chloroplasts is via binary fission, where they just duplicate the DNA, the circular chromosome, and then they split. We call that binary fission. And remember that prokaryotes also undergo binary fission, and mitochondria are similar in size to bacteria. So those are the things that support the endosymbiotic theory. Okay. All right. Enough of that. Mitochondrial DNA. So mitochondria has its own DNA, and you inherit your mitochondria from your mother, right? In this little illustration here, mitochondrial DNA inheritance pattern, it comes by way of your mother. Okay. And so that's where the idea of the mitochondrial Eve comes from. Eve being the first woman to ever exist, right? Because we can trace your mitochondrial, we can trace you back by way of your mitochondrial DNA, back to your mother, back to your grandmother, back to your great-great-grandmother, and so on and so on and so on. And hypothetically, we can go all the way back to the first woman that ever existed by way of mitochondrial DNA. Because everybody's mitochondrial DNA comes by way of the female, of the mother's line. So you inherit your mitochondrial DNA from your mother, and she got it from her mother, and so on and so on and so on, back to the first woman that ever existed. We call her mitochondrial Eve, the matrilineal, most recent common ancestor of all living humans. So she's defined as the most recent woman from whom all living humans descend in an unbroken line by way of mitochondrial DNA. Again, it's hypothetical, however, well, I should say theoretical, because the evidence supports that idea. Okay, and that's the end of, I didn't realize, that's the end of chapter 4. And there you go. So next up, chapter 5.